Hello and welcome back to the Biochemistry for Health Sciences channel. We'll continue with the regulation of metabolism during starvation, and in today's video we shall look at the effects of glucagon on the body. In the previous video we mentioned why studying glucagon physiology has been very challenging. Some of the reasons include unreliable assays for this hormone, complex regulation of glucagon secretion, species differences. A lot of research is done in rats, but that information may not apply to humans. Glucagon has a very short half-life, so it doesn't stay around in the blood long enough for us to analyze it. Glucagon interacts with other receptors, and that depends on the dose of glucagon that is given. Then there is the direct versus indirect actions that is mediated by the sympathetic nervous system that can affect glucagon secretion. So we'll focus on the well-established effects of glucagon that we know. Glucagon affects mostly the liver. So far, we have not detected glucagon receptors in human adipocytes or human muscle cells. So we'll focus this discussion mainly on the effects of glucagon on the liver. During starvation, the alpha cells in the pancreas will secrete glucagon, and this glucagon will now bind to receptors, glucagon receptors found on the liver cells. These glucagon receptors, GCGRs, these receptors belong to the G protein coupled receptor family, GPCR. And these receptors are very closely related or associated with a protein called G-alpha-S, a stimulatory G protein. Now normally this G-alpha-S, and you can see this protein here, G-alpha-S, very closely associated with the glucagon receptor. So under normal conditions, G-alpha-S has GDP nucleotide. The nucleotide bound to this protein is a GDP. However, once glucagon binds to the receptor shape changes, the receptor conformation changes, and that leads to the replacement of GDP with GTP, because this protein is very closely related to the receptor. The G-alpha protein conformation also changes, and that change in this G-alpha protein will cause the GTP to be kicked out and replaced by GTP. So now the G-alpha protein has GTP bound, and when it has GTP bound, it becomes activated. Now it's important to note that the G-alpha protein also has GTPase activity. So it slowly can break down the GTP that is bound onto that protein. It slowly can break it down or hydrolyze it back into GDP. And once it converts into GDP, then once again the protein can become inactive. So this cycle can keep on happening, GDP binding or being replaced by GTP, and then the GTP slowly being hydrolyzed to give you back the inactive GDP protein. However, once the GTP is bound and before it gets hydrolyzed, this active GTP-bound G-alpha protein will now go and bind another protein, and this is an enzyme found on the membrane and this enzyme is called adenyl cyclase. Now this enzyme, when it binds to G-alpha S, becomes activated. It becomes an active enzyme. And what does it do? The enzyme will now convert ATP into cyclic AMP. So adenylyl cyclase will catalyze this reaction. So as ATP gets converted into cyclic AMP, the concentration of cyclic AMP in the cell increases. Now, cyclic AMP is considered an intracellular hormone or a second messenger. Of course, the first messenger is glucagon. So as the concentration of cyclic AMP in the cell increases, the cyclic AMP will now bind to these sites on a protein called protein kinase A. These are the regulatory sites or the R sites. And when cyclic AMP binds to the R sites of protein kinase A, that will allosterically activate the catalytic sites of protein kinase A. So now protein kinase A becomes an active protein. Now protein kinase A can be attached to different parts of the cell. So in this case, we see the protein kinase A attached to the cell membrane part, near the cell membrane, near adenyl cyclase. And this attachment of protein kinase A is done by proteins called A-anchoring proteins or AKAPS. So in this case, we see the pKa molecule held very close to the adenyl cyclase near the plasma membrane by this AKAP protein, this A-anchoring protein. So to summarize, so we have the signal glucagon that is released during starvation, and the signals will bind glucagon receptors on the liver cells, and the G-alpha protein that is associated with this receptor will become activated by replacing the GTP by GTP. And once the G-alpha protein becomes activated, it will go and activate the enzyme adenyl cyclase, and this enzyme will now convert ATP into cyclic AMP. And the cyclic AMP will allosterically activate another protein that is held very close to the adenyl cyclase, and this protein is called CAMP-dependent protein kinase A, or simply pKa. So now we have pKa that has become very active. So what happens next? One of the major effects of glucagon on the liver is that glucagon increases the breakdown of glycogen or glycogenolysis, while at the same time decreasing the synthesis of glycogen or glycogenesis. And this happens by stimulating the key enzyme for glycogen breakdown, which is glycogen phosphorylase, and inactivating or inhibiting the key enzyme for glycogen synthesis, which is glycogen synthase. So activate this enzyme, inhibit this enzyme. Let's see how glucagon does this. Now, in case of glycogen synthase, the key enzyme involved in synthesis of glycogen, the active form or the R form is the non-phosphorylated form. The phosphorylated form is the inactive form or the T form. So what pKa does, now pKa, remember, has become very active, this protein kinase A. Protein kinase A phosphorylates, adds phosphate groups to the glycogen synthase, and so the glycogen synthase becomes phosphorylated, becomes the T or the inactive form, and that will slow down or inhibit glycogen synthesis. In case of glycogen breakdown, the key enzyme is glycogen phosphorylase or PYGL. Now the inactive PYGL is the unphosphorylated form, while the active PYGL is the phosphorylated form. So this conversion of unphosphorylated form into phosphorylated form is carried out by this enzyme PHK, which is phosphorylase kinase. So phosphorylase kinase will catalyze the phosphorylation of glycogen phosphorylase into the active glycogen phosphorylase, and when this glycogen phosphorylase becomes active, that will lead to increased glycogen breakdown. Now, phosphorylase kinase also exists as the unphosphorylated form, which is inactive, and the active phosphorylated form. So what does pKa do? pKa phosphorylates phosphorylase kinase, phosphorylates this enzyme into the phosphorylated form. And now this enzyme becomes active. So pKa makes this phosphorylase kinase active, and now this kinase will phosphorylate the inactive glycogen phosphorylase into the active glycogen phosphorylase, and that will lead to increased glycogen breakdown. Another major effect of glucagon on the liver is that glucagon decreases glycolysis in the liver while it increases gluconeogenesis, the synthesis of new glucose. So remember, we discussed this in previous videos. Gluconeogenesis is the synthesis of glucose, which happens mostly in the liver, while glycolysis is the breakdown of glucose. So let's see how glucagon decreases glycolysis in the liver while increasing gluconeogenesis. Now, one way by which glucagon increases gluconeogenesis is actually increasing the amount of gluconeogenesis enzymes. So pKa, which is now activated, will go and phosphorylate this transcription factor called CAMP response element binding, or CREB. Once CREB becomes phosphorylated, it becomes activated. This transcription factor will now go and bind to the DNA of the genes that are responsible for the synthesis of key gluconeogenesis enzymes such as glucose 6-phosphatase as well as PP carboxykinase. So here is PP carboxykinase, here is glucose 6-phosphatase, two of the enzymes that are very important for or unique for gluconeogenesis. And the amount of these enzymes will increase. So when the amount of the enzyme increases, you see the reaction rate will start increasing, and that will lead to increased activity of these enzymes, and that will lead to increased gluconeogenesis. Now, it's important to note that during starvation, the levels of insulin goes down. So without insulin, as you remember in the previous video on insulin, there is no more blockades on gluconeogenesis by the FOXO1 transcription factor. And this is something we discussed in video 6.10. So now FOXO1 is another transcription factor that can enter the nucleus, bind to the gluconeogenesis genes, and those genes will be transcribed into mRNA, which eventually will lead to the synthesis of more gluconeogenesis enzymes, and that will also lead to increased gluconeogenesis in the liver. Another effect of glucagon is that it decreases the concentration of this chemical here, fructose 2,6-bisphosphate. So how does this happen? Well, notice that we have a bifunctional enzyme called phosphofructokinase 2 fructose 2,6-bisphosphatase 2, or PFK2 FBP2. So this is a bifunctional enzyme. Now, when this enzyme is non-phosphorylated, this part FBP2 is inactive while the PFK2 part is active. However, when it becomes phosphorylated, and this is done by the active pKa, so during starvation, glucagon is released, causes the activation of pKa, this bifunctional enzyme gets phosphorylated, and now the PFK2 activity is inactive, so this direction does not happen, while the FBP2 activity becomes active. The fructose 2,6-bisphosphate is converted into fructose 6-phosphate. So what happens? The concentration of fructose 2,6-bisphosphate in the liver cells start going down and down and down. Now, fructose 2,6-bisphosphate is a very strong activator of the glycolysis enzyme phosphofructokinase 1. This enzyme that catalyzes this reaction here, where fructose 6-phosphate is converted to fructose 1,6-bisphosphate. So this enzyme is activated by fructose 2,6-bisphosphate. But now as the fructose 2,6-bisphosphate concentration goes down, this activation goes down. That means PFK1 activity decreases, and that will decrease glycolysis. So this decrease in concentration of fructose 2,6-bisphosphate is going to decrease glycolysis in the liver cells. Now, another effect of fructose 2,6-bisphosphate is that it is a strong inhibitor of fructose 1,6-bisphosphatase, this enzyme that is required for gluconeogenesis. And now that this inhibition does not happen, now gluconeogenesis starts increasing. So gluconeogenesis starts increasing in the liver. So fructose 2,6-bisphosphate concentration going down will lead to increased gluconeogenesis and decreased glycolysis. Yet another effect of glucagon is that it decreases glycolysis by phosphorylating and inactivating the enzyme pyruvate kinase. So pyruvate kinase, which is the enzyme that catalyzes this reaction 10 of glycolysis, where PP becomes pyruvate. This enzyme, when it's in the non-phosphorylated form, it is active, but when it's in the phosphorylated form, it becomes inactive. So pKa will phosphate pyruvate kinase, make it into the inactive form, and that means this reaction will not happen. Phosphoenol pyruvate will not be converted to pyruvate, glycolysis slows down, and phosphoenol pyruvate will accumulate, and this phosphoenol pyruvate will enter gluconeogenesis. Okay, so PP will accumulate and will now enter gluconeogenesis, and with more PP, gluconeogenesis increases while glycolysis is inhibited. Now, another effect of glucagon is that it phosphorylates and inactivates the enzyme pyruvate dehydrogenase. Remember, pyruvate dehydrogenase is a mitochondrial enzyme that converts pyruvate into acetyl-CoA. So pyruvate can come not only from glycolysis, but remember glycolysis is being inhibited, but pyruvate in case of starvation in the liver, So pyruvate can also come from other sources besides glycolysis. It can come from lactate, it can come from various glucogenic amino acids. And so what happens is this pyruvate that comes from other sources, instead of being converted to acetyl-CoA, it is sent into gluconeogenesis, because this reaction here carried out by PDAs is inhibited. So what happens is pKa will phosphorylate the unphosphorated PDH, which is the active form, into the phosphorated pyruvate dehydrogenase, which is the inactive form. That will lead to the inhibition of this reaction. Pyruvate will not be converted to acetyl-CoA. Instead, pyruvate will be sent into gluconeogenesis. So the liver will increase gluconeogenesis. Whatever pyruvate it gets from other sources will enter gluconeogenesis. So to summarize, glucagon during starvation will cause the liver to break down glycogen to glucose, and then it will also increase synthesis of glucose, and so the liver will now start producing quite a bit of glucose during starvation, and this glucose will be sent out into the bloodstream to maintain blood glucose levels. And remember, this glucose is very important for the brain and the red blood cells that rely very heavily on glucose for their energy needs. It should be remembered that during starvation, glucagon is not the only hormone that affects blood glucose levels. There are several other hormones such as epinephrine, cortisol, growth hormone, as well as thyroid hormones that also will affect blood glucose during starvation, and we'll talk about some of these effects, some of these hormones in future videos. So besides increasing glycogen breakdown and gluconeogenesis in the liver, another effect of glucagon is that it increases lipid breakdown in the liver. Let's see how this happens. Now, pKa that becomes activated when glucagon binds to the receptors on the liver cells, as we mentioned earlier, will phosphorylate the transcription factor CRAB. And when CRAB becomes phosphorated, it becomes activated. Now, this will bind to the gene for the enzyme carnitine acyltransferase 1 or CPT1. And this enzyme plays a very important role in the transport of fatty acids from the cytoplasm into the mitochondria, where the fatty acid will be degraded by beta oxidation. So by increasing the amount of this enzyme, more fatty acids from the liver cell cytoplasm will start entering the mitochondria, and there the fatty acids will be broken down by beta oxidation. So besides increasing the transport of fatty acids into the mitochondria for breakdown of the fatty acids by beta oxidation, another effect of glucagon, what pKa does, is that it will phosphorylate a very important enzyme for lipid synthesis, for fatty acid synthesis, and that enzyme is acetyl-CoA carboxylase, or ACC. So pKa will make active ACC into the inactive ACC that is phosphorylated. So this step will not happen. Acetyl-CoA will not be converted to malonyl-CoA, and without malonyl-CoA, the fatty acid will not be synthesized, so there will be less triglyceride synthesis and therefore less release of VLDL by the liver cells into the bloodstream. And this also means the concentration of malonyl-CoA is very low, okay, because this step is being inhibited. And since there's no malonyl-CoA, remember normally malonyl-CoA will inhibit CPT1, this inhibition is gone. Okay, so that leaves CPT1 to be very active, and that will cause the fatty acids to enter the liver cells and become broken down by beta oxidation. So this is how glucagon can increase lipid breakdown. Another effect of glucagon that has been reported is it increases beta oxidation by increasing the transcription of CPT1 as well as the enzyme ACOX1 or acyl-CoA oxidase 1, which is the important enzyme for beta oxidation in the peroxisome. And this happens because glucagon increases the AMP to ATP ratio, and that causes the activation of the enzyme AMP activated kinase or AMPK. And this AMPK will cause the activation of another transcription factor, and this transcription factor is called peroxisome proliferator activated receptor alpha, or PPAR alpha. And when this transcription factor is activated, it leads to the increased gene expression, and that increases the amount of CPT1 as well as ACOX1. CPT1 will cause increased beta oxidation in the mitochondria, while ACOX1 will cause increased beta oxidation in the peroxisome. Another effect of glucagon is that it increases the LDL receptor activity on liver cells. So there is an inhibitor called proprotein convertase subtlysin casein type 9. This inhibitor, so this is an inhibitor of the LDL receptor. Okay, so this inhibits LDL receptors. And what glucagon does is that it increases the degradation of this inhibitor. Increases the degradation of this inhibitor of the LDL receptors, which means that will lead to an increase in the functional LDL receptor activity of the liver cells. So with this increased LDL receptor activity, what's going to happen is it's going to take in more cholesterol into the liver. Okay, so that's another effect of glucagon. It increases the cholesterol uptake into the liver by increasing the activity of functional LDL receptors. Glucagon also affects amino acid metabolism. Glucagon actually increases the catabolism of amino acids in the liver. And that's important because amino acids supply the carbon skeletons for glucosynthesis, for gluconeogenesis. So how does this happen? How does glucagon increase amino acid catabolism? Let's look at that. So glucagon, as we mentioned earlier, increases CRAB activity, and the activated phosphorylated CRAB activity leads to the production of more amino acid transport proteins. So more of these amino acid transport proteins are made by the liver, which means more amino acids are transported into the liver, as well as the urea cycle enzymes. So remember, the nitrogen part of the amino acids is converted into urea, so these urea cycle enzymes also increase during starvation by glucagon. And this is, as I mentioned earlier, important because these amino acids will provide, so amino acids like your alanine, glutamine, they will provide the carbon skeletons, for example, oxaloacetate, that is necessary for the liver to make glucose by gluconeogenesis, and that glucose can be sent out into the bloodstream during starvation to maintain blood glucose levels. So by transporting more amino acids and catabolizing these amino acids, the liver will be able to make more glucose by gluconeogenesis. So this is an important relationship, what we call the liver alpha cell axis. So a relationship between the liver and the alpha cells of the pancreas. And this axis is basically a physiological feedback loop where several different amino acids will stimulate glucagon secretion by the alpha cells. And these amino acids are called gluconotropic amino acids. The glucagon that is released because of this amino acid stimulation in turn will increase the amino acid uptake and metabolism by the liver. Okay, so what we talked about earlier, gluconeogenesis and ureagenesis is increased by the liver. So this relationship between amino acids and glucagon is referred to as the liver alpha cell axis. So this is basically what we know today is the effect of glucagon on the liver. Now, as we mentioned earlier, glucagon does not affect adipocytes, although in rat we do see glucagon receptor mRNA that is present, but in case of humans, that has not been reported yet. No glucagon receptors have been clearly established in human adipocytes. Now, some researchers have given very high glucagon doses and have seen lipolysis in adipocytes, but this may actually be due to release of catecholamines when we use high glucagon doses. So this lipolysis that they saw in adipocytes may not be a direct effect of glucagon. It may be indirectly caused by catecholamines. So the lipolysis that we talked about that happens in adipocytes during starvation may mostly be due to the fact that the insulin levels are low and not because glucagon levels are high. So just like adipocytes, no glucagon receptors have been found in human muscle cells. So the proteolysis that we see in muscles during starvation may be due more to the effects of cortisol as well as maybe some catecholamines, but is not due to glucagon. So to summarize, glucagon affects mostly the liver, not the adipocytes or the muscle cells. And in the liver, it basically increases glycogen breakdown, glycogenolysis, increases gluconeogenesis, and these two effects means liver is producing more glucose, and that glucose will help maintain the blood glucose concentration during starvation. It will also increases fatty acid breakdown, and it increases amino acid catabolism, and this amino acid catabolism will release carbon skeletons, and that carbon skeletons is important for making glucose by gluconeogenesis. So that's what glucagon does, mostly affects the liver. In the next video, we'll talk more about other hormones that are important in starvation. So until then, once again, thank you very much for your support as well as watching this video. Until next time, have a wonderful day and we'll see you soon.